food has entered into our duodenum, okay, and food has arrived from this amazing organ called stomach into our duodenum, right? This is your oh my gosh, my diagram just went away. Okay, so food is in the duodenum right here, and here is your neighboring, loving neighboring pancreas. And pancreas is like, oh, we have some neighbor here, some amino acids, some fatty acids. Cool. So pancreas is amazing hormone. Sorry, amazing gland. It has both endocrine and exocrine function. Okay, it has it can make hormones and produce it outside, and it can just from within inside, right? So let's talk a little bit about this naming: exocrine, endocrine, and paracrine. This is a very fundamental concept. So endocrine, what is endocrine? So simple definition is that hormones are released inside. Endo means inside the bloodstream, and they travel to their target organ, right? So that's like endo, it means within. Example is like insulin from beta cells of pancreas. They travel through blood, act on muscles and liver, right, to lower this glucose, right? Paracrine. Now, definition is that these paracrine, it means it's a chemical messenger, right? And it acts locally and it's released locally, right? It is released from the neighboring cell and it acts on the neighboring cell, right? And it is done in the same tissue. It does not enter in the bloodstream. So that's very important. So paracrine, around, right? Para means beside or around, right? So that's, remember. Exo, amazing. Exo means outside, right? So how? So secretion of hormones or chemicals are released into the ducts that lead to an external surface like digestive tract or skin. Okay, so important thing here is that exocrine means duct, exo means exit. So exit is usually done via some duct. So key point here is that exocrine system has ducts and the product goes outside the body or into a lumen. Okay, example, again, I'll give you pancreas. Here we will read about acinar cells, and usually whenever we read about acinar means this exocrine. Okay, saliva is also produced by acinar cells, right? So these acinar cells, they secrete these big gigantic digestive enzymes called amylase, lipase, trypsinogen, blah 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 blah, into something called as pancreatic duct. Now this pancreatic duct is connected to the duodenum. Okay, now food is coming from stomach into duodenum, and pancreas releases these pancreatic enzymes inside pancreatic duct. So there is duct. So where there is duct, where there is a duct, there is exocrine. So exo means outside. So I hope we understand, the naming. I mean, okay. Okay, so usually endocrine is like ductless, exocrine is duct. Usually that's the story. Okay, so we have this thing. We have this small intestine, this duodenum, this is pancreas, right? We have common bile duct that coming right here. We have pancreatic duct, the whole duct, right? Hormones released inside the duct is exocrine. Tail of pancreas, here are the beta cells, right? And this is the lobule, right? And these lobules have acinar cells that secrete digestive enzymes, and these digestive enzymes are just trypsinogen, pepsinogen, blah blah blah, and gin. They produce a lot of enzymes that can break amino acids. So that's the concept of pancreas right here. And these exocrine cells, they secrete pancreatic juice. What is pancreatic juice? Pancreatic juice is just enzymes. So they can digest all nutrients. Okay, that's very important. Conceptually, it can digest all nutrients. It should be a nutrient. It should not be a coin. It should not be a toy. It's not going to digest a toy. That's important. How much? 1500 ml, a liter and a half. pH, very basic in nature. 8.3. Okay, very important, 8.3. Now why the pH is 8.3 right here? Because pancreas, this pancreatic system, this bicarb system works via secretin. Okay, secretin, we'll talk about it. Why the pH is 8.3 here? Why? So the function of these secretory pancreatic juice, the pancreatic juice here, this is digestion of fats, digestion of protein, and a little bit digestion of carbs. Mostly carbs are digested by saliva, and it's least digested by stomach. That's very important. Fats are digested by pancreatic lipases. Okay, we have three type of lipases. We have salivary lipase, we have this stomach lipase, that's called gastric lipase, and then we have pancreatic lipase. And these lipases are helped or assisted by these co-lipases. Now, don't think this co-lipases is just like a, it's like a Robin to a Batman. No, actually, it's opposite. This co-lipase is the Batman, and lipase is the Robin here. Okay, so that's very important. I will tell you what's the function of this co-lipase a little bit later. Protein is digested by these thing called zymogens. Now, zymogens are these inactive enzymes. Why they are inactive? Because they are dangerous as hell. If they get activated in serum, in your blood, they will digest away all the protein that is binded to your essential compounds like calcium, like T3, like these things, right? So, what are these zymogens? We have three type. We have one of the main type is trypsinogen. Then we have these chymotrypsinogen. Then we have para-carboxypeptidase, right? So we have these three: trypsinogen, chymotrypsinogen, and pro-carboxypeptidase, not para. So we have these three things. Let's talk a little bit about these things, right? Story of these zymogens. So this zymogen called trypsinogen gets activated in the presence of enterokinase or peptidase. Now, the initial trigger for everything is this enteropeptidase or enterokinase. Okay, kinase means it puts one PO4 on. I mean, it's how the ATP usually travels, right? It's called phosphorylation. Okay, it's called phosphorylation. It just joins one PO4 on the on the thing that is happening. It comes this enterokinase or peptidase. It originates, not comes. I have to stop writing comes. So it originates from duodenal brushes border in small intestine. So duodenal again, very important. So once food is there and this duodenal brush border senses, okay, we have some amino acids right here. It releases this called enterokinase. Now once this is released, it acts on trypsinogen and it converts trypsinogen into trypsin, right? It's a kinase. And after this happens, this trypsin goes and it activates pro-carboxypeptidase into carboxypeptidase. Pro means before, so right, carboxypeptidase before it was pro-carboxypeptidase. So smart. Then trypsin also activates chymotrypsin into chymotrypsin. Okay, so we have these active compounds, these chymotrypsinogen, trypsin, and carboxypeptidase. Now, all of these three are responsible for protein digestion. That's the end of the story. Okay, cool. What about the story of fats, right? Digestion of fat. I told you pancreas lipase is not the main hero. The main hero is co-lipase. Let's talk from start. So this is the story of co-lipase. The co-lipase, so fat, enters as fat globules in our food, right? Via our food. Now, they are hydrophobic in nature because it's a fat. Have you ever tried mixing fat with water? It never works, right? Try olive oil. I don't know. Maybe you don't have olive oil. Okay, just add some refined oil with water, and then it will not mix because it's hydrophobic in nature. Now, these hydrophobic fat globules, what should I do with them? What happened here is that we have these lipases that are produced in pancreas, right? These lipases are lipophilic. It means they're fat lovers, right? So weird when I say this, I am a fat lover. So lipases are lipophilic, and this lipase, they go and they bind with these fat globules and they love them. And lipase can't reach fat surface now, right? Lipase, right, can't reach fat surface. So what happens is that here that bile salt comes and it acts as a detergent and it emulsifies fats into small fat droplets. So we had big fat and now we have small fat here, right? And what happened here is that this actually leads to increased area for lipases to work on. Okay, so now lipase can go and act like love it because because it has more surface. But imagine we have big fat molecules, we have teeny tiny lipase. Can it bind? No. So we have this this little bit of lipase is coming and loving every this little bit of small fat droplets. So increased surface area, good, good lipase story. But here's the paradox. Paradox is that bile has, two type of ends. One is a hydrophobic end, one is a hydrophilic end. And hydrophobic is inside and hydrophobic is outside. So hydrophobic means it's not gonna, it's not gonna dissolve in water. And hydrophilic is it's gonna dissolve in water. So bile coats so strong that, and it coats so effectively that it blocks the pancreatic lipase. And we know that pancreatic lipase is a fat lover. Okay, so fat, now this lipase, this pancreatic lipase cannot love this fat globules, right? So lipase like, okay, I'm gonna coat this. So bile coats so much, so strongly, this lipase, it cannot bind with these fat globules. So what happens here? So colipase comes and it, enter colipase in the chat. Okay, so colipase comes and it binds to both lipase and fat droplet surface. So we have this lipase, right? We have this fat droplet. It is coated by one hydrophilic and hydrophobic, right? So we have this hydrophobic side and hydrophilic. So this is the hydrophilic, this is hydrophobic, right? And this lipase, it's binded to this fat surface with the help of colipase here, right? This is a colipase, and it's binded with each other. So this colipase acts as an anchor molecule here. And so what happens after this is that, so pancreatic lipase can work now. So without colipase, pancreatic lipase is useless. So that's why I was saying that colipase is main guy here. Note, inflammatory processes, right, can result in premature activation of zymogen. I told you zymogen is dangerous. So zymogen gets activated, and once zymogen is activated, trypsinogen gets activated, and then trypsinogen creates this trypsin. Trypsin creates this carboxyl peptidase, and carmopeptidase are working now. What happened here is that then it leads to acute pancreatitis. And what happens is this, that this pancreas, which was storing, which has this storage of these dangerous zymogen, they get activated and it digests itself. And then we have something called as liquefactive necrosis, right? It's a liquid. Okay, if we open the pancreas, it's a liquid. Okay, so usually pancreatitis can be of two types: a pancreatitis of the borders of the outer surface of pancreas, and a pancreatitis of inside, right? So we have liquefactive necrosis right here. Okay, regulation of what? Of these pancreatic juices. So regulation is done by, okay, something called as cholecystokinin pancreozymin. Okay, cholecystokinin pancreozymin, this is called CCKBZ, and it leads to constriction of gallbladder. Like it constricts, like it contracts, constriction. I hope you understand this. And it increases the pancreatic release, secretion of pancreatic enzyme outside, right? It just constricts this, right? So first is this is first hormone. Second hormone is called secretin. Now this is a nature's antacid, right? What happened? I told you why we have this basic, this is the reason. Because we have something called as secretin, and these secretin are released by S cells, and these S cells are present in the duodenum, and they make secretin. Secretin is released in the intestine, and it stimulated by acid chyme, whatever you want to call. So S cells is released due to acid, presence of acid. So secretin is released. It leads to releasing of bicarb. Now, where is this bicarb coming from? This bicarb is coming from this CO2 plus H2O gives, H2CO3. We have one H plus and one bicarb, right? It is done by a carbonic anhydrase. So this whole, the same way acid is produced is the same way bicarb is produced. Usually the theme in the body is that if there is some production of acid, there will be some production of base simultaneously. So body always try to make sure that you are running on equilibrium. That's very beautiful. So we have bicarb, and it goes inside your duodenum to suppress this acid that just arrived. I have to stop using this word come. So this acid that just arrived from stomach into duodenum. Okay, so bicarb does that. It leads to alkalinity of pancreatic secretion for the acid neutralization. I hope we understand this. So, note, okay, secretin, first hormone to be discovered. Amazing, amazing point. Decreases the enzyme concentration, right? Okay, so we understand this. Let's talk about mechanism of bicarb. How bicarb is produced inside the lumen. Okay, so this is one pancreatic cell. This is the lumen of bicarb, and the pH here is around 8.3. Here, I told you. Here, this pH 8.3, this pH. So why the pH is here 8.3? How? So I told you we have carbon dioxide plus water under the influence of, not influence, this is a catalyst. It's called carbonic anhydrase. Now this is catalyst used, and we get something called as H2CO3. Now this H2CO3 makes one molecule of proton, that is H plus, and another molecule of bicarb, that is base. And this is acid, this is base. Now this base is pushed inside the lumen with the help of this channel. This is an exchanger. It means it exchanges. It's not active. How can it exchange? Because here is low concentration of bicarb. Here is high concentration of bicarb. Here is low concentration of bicarb. So it just pushes. And in this way, it also gets one chloride ion, negatively charged. So it gets this. And where is the chloride coming from? This chloride is it's okay. The origin of this chloride is from something called as CFTR channels. CFTR, cystic fibrosis transmembrane conductance regulator. Now this is a chloride channel opening, and if we have something mutation in this, we will not be able to make chloride. We will not be able to produce chloride inside this lumen. If we have less chloride, we will not be able to use this exchanger. If we have less bad exchanger, we will not be able to produce bicarb inside the lumen. And then the pH of pancreatic, the pH of pancreatic juice will drop from 8.3 to, I don't know, maybe 7, and which is very bad for you, because now we have this acid that is coming from stomach and that's destroying your duodenum, and now you have bad base. We have weak base, which is because of mutation in the CFTR, cystic fibrosis transmembrane conductance receptors. Now these are ATP gated or ATP sensitive. Okay, here's the thing. They are ATP gated, right? And they are ATP sensitive. Cool. Now they are passive channels. Okay. Now they are ATP sensitive. Okay. They are passive channels. It means they work passively. It means they work on the concentration gradient. Here was a lot of chloride already, and then they've just pushed it. Okay. So passive channels. ATP is used for the regulation of these channels. I should remove this because this is a little bit confusing word right there. So that's the story of this, this CFTR. I hope we understand this topic. So that was, that was pancreas for you, but exocrine pancreas.